This Week in Microbiology is brought to you by the American Society for Microbiology at microbeworld.org/TWiM. From Microbe TV, this is TWiM, This Week in Microbiology, episode 132, recorded on August 4th, 2016. This episode is brought to you by CuriosityStream, a subscription streaming service that offers over 1,500 documentaries and nonfiction series from the world's best filmmakers. Get unlimited access starting at just $2.99 a month. And for our audience, the first 2 months are completely free if you sign up at curiositystream.com/microbe and use the promo code microbe. This episode is also brought to you by Drobo, a family of safe, expandable, yet simple-to-use storage arrays. Drobos are designed to protect your important data forever. Visit drobo.com to learn more. Listeners can save $100 on a Drobo system at drobostore.com by using the discount code microbe100. Hello, everyone. I'm Vincent Racaniello, and you are listening to TWiM, the podcast that explores unseen life on Earth. Joining me today from Small Things Considered, Elio Schaechter.
Hey, how are you doing? Today that we're recording is kind of a memorable date. It is the 4th day of the 8th month of the 16th year.
Wow.
So it's 2 to the 4th, to the 3rd power, and to the 4th power.
That's nice. That is a memorable day.
Wow.
Is there a name for that? No,
I don't know.
It's not like a mole
Let's
day.
make one up.
You know the mole day, which is Avogadro's number day.
Right.
It's not that one. Also joining us from Ann Arbor, Michigan, Michele Swanson.
Hello.
We were last united at ASM Microbe in Boston.
That's right.
Which seems so long ago. It was in June, which is at the beginning of summer.
And it was action-packed, wasn't it?
It was a lot of fun. Yeah, I had a good time. It was a great meeting. What did you think? Good meeting?
Yeah. I mean, it was broader, right? So there were
yes, for sure.
The science was covered a wider range, and it seemed like there are a lot of other activities going on as well.
Yeah.
Like kind of community building activities.
Yeah, I agree. I thought I also ended up in a lot of clinically oriented talks, which was fine. Mm-hmm. And that, of course, was the combination. I did enjoy Bill Gates, though. That was like...
What surprised me is that the convention center in Boston is so huge.
It is
that I didn't feel it was crowded.
Yes, it is. It's a huge place. I must say that I can barely remember it. It seems so far in the past.
Between Microbe and now, I was also able to attend ASMQ, mm-hmm. which is the ASM-sponsored meeting for undergraduate educators, and that was terrific. It's a smaller, much smaller than Microbe, but the people there are just really energized and committed to education, and they have great ideas that they want to share with their colleagues. So
isn't that nice?
Yeah, it's a good meeting.
And at both places, I just want to say to the listeners who I was able to meet at both Microbe and ASMQ, thanks so much for your encouraging words and ideas, and keep them coming.
Mm-hmm. Yeah, I think ASMQ is fun. I went one year also, and I think it's really interesting, different perspective, and
mm-hmm.
a lot of fun. Good meeting. Support ASM. Go join. All right, today we have a snippet and a paper for you. And I'm going to do the snippet, which is a paper in MBio. It is called Cell Division by Longitudinal Scission in the Insect Endosymbiont Spiroplasma pulsoni. And it is by Elodie Ramon, Catherine McLaughlin, Stephanie Clerc-Rosset, Graham Knott, and Bruno Lemaitre. It's from the École Polytechnique Fédérale de Lausanne, which, of course, is in Lausanne, Switzerland. This caught my eye. Perfect snippet. It's all about an unusual way that some bacterial cells divide. A bacteria called Spiroplasma. These are helical bacteria, very motile. They have no cell wall. They're found in...
They're cousins of the mycoplasma, aren't they?
Cousins
they belong
of
to
the
mollicutes.
Mollicutes. That's right. Mollicutes. I didn't say that because I wasn't sure how to pronounce it. Mollicutes. They are found in 5 to 10% of all insect species. Some of them are horizontally transmitted. They can be pathogens or they could be gut commensals. And others are transmitted vertically from mother to offspring. And the one in this paper is called Spiroplasma poulsoni. It's vertically transmitted. And together with Wolbachia, these are two natural known symbionts of Drosophila melanogaster. And many of you probably have heard of Wolbachia. It's the best studied, I think, of these insect endosymbionts. And also very famous recently because its presence prevents multiplication of some viruses, including flaviviruses like Zika virus. So putting Wolbachia into mosquitoes is one idea that people have for inhibiting transmission of viruses. Spiroplasma poulsoni is mostly present in the fly in the hemolymph. This is the circulatory fluid, which would, I guess, be equivalent to blood in humans. And it's not detected by the immune system of the fly. It's not detected. It's not cleared or affected in any way by the immune system. But it does have an effect on the host, very much like that of Wolbachia. It can affect the sex ratio. It kills males at early embryonic time points. All right, so during the embryonic development, the males are killed, so you mostly have females that are born, so it affects the sex ratio. And that property, together with protecting the fly against infection with some parasites, are thought to maintain the endosymbiont in the fly population. So it gives it a selection, so it stays there. Now, this group that I mentioned at the École Polytechnique, they have been studying the biology of Spiroplasma poulsoni because we don't know much about it. And that's where the work in this paper starts. And what they do is they start out by simply staining the bacteria with a fluorescent DNA stain. And you can see it's a typical helical linear bacterium, but they also found in their cultures Y-shaped bacteria as well. Now
why?
Why? That's the question. You were waiting for that, right? Why are they Y-shaped? That's a good question. So the DNA stains the DNA. You can see throughout the spiral bacteria. It's also in both arms of the Y. And they said, well, this could be simply bacteria getting twisted around each other. So to rule that out, they did transmission electron microscopy, very high magnification. You can see that the cytoplasm is continuous between the two arms of the Y and the trunk. So the Y is made up of two arms, and the trunk is the single structure at the bottom. And the cytoplasm is continuous. So it's not just two bacteria twisted around each other. So they think, well, these must be dividing cells. So what do we know and how can we prove this? Now, in bacteria, most bacteria, the cell divides by a process called cytokinesis, the division of the cytoplasm. And an important protein in that process is a protein, it happens to be a GTPase, a protein that hydrolyzes GTP, called FTSZ or FTZ, filamenting temperature-sensitive Z protein. We've mentioned it before on TWiM. This protein assembles on the inner face of the bacterial membrane. It makes what's called a Z ring, and then it recruits other proteins that provide the contractile force for pinching the membrane and having the scission event that breaks the two parts of the cell in half. So they said, are there FTZ proteins in this bacterium? And in fact, there's one gene encoding a FTZ protein. So they get an antibody to it. They stain the bacteria and ask, where is the FTZ protein? And they see the protein at the only on the Y forms. They don't see this FTZ protein on the linear forms. And they see it where the arms are coming together to the trunk. So if that's in fact the division point, it would make sense if a FTZ protein is there. And then they do another series of experiments where they use inhibitors, drug inhibitors of cell division, to ask what's this Y, what's the function of the Y. So they have an inhibitor of the GTPase that's part of FTZ. It's very convenient. And this inhibitor blocks cell division in other bacteria. So they inject it into the flies, and then they pull out the spiroplasma and ask what's happening. And when they use this inhibitor of the GTPase, it causes aberrant Y morphology. So the Ys are not Ys anymore. One arm is a little bit shorter. So they've somehow affected this process. Yeah.
I thought that was a clever use of that reagent.
Very good.
Whose mechanism is well known because they probably don't have genetics in this organism and they wanted to come up with alternate mechanisms. So I found that elegant.
They can't even cultivate it, can they?
No, I have to put it in flies, right? Yeah. That's why they had to put the drug into flies because they can't grow it in medium or on plates, yeah. Right.
And they have lovely transmission EMs, but then also the quantitative data to bolster their image selection. So
another cell division inhibitor they use is ciprofloxacin, a well-known antimicrobial, which is an inhibitor of DNA gyrase, and therefore it's an inhibitor of DNA replication. And this causes, when added to the flies, it causes abnormally shaped spiroplasma with multiple branches. It's not a Y anymore. There's just multiple branches off of the main trunk. So both of these inhibitors are affecting the nice Y formation, and they also inhibit growth of the bacteria, which would be consistent with inhibiting division, of course. Now, as I mentioned before, they did an experiment to stain these bacteria with a fluorescent stain against DNA,
and gold stain.
This is DNA now. We're looking at DNA again.
Oh, sorry.
The gold was for the protein, right?
Right.
They stain, when they look for DNA with a fluorescent stain, they see the DNA is in both arms and trunk of the Y. If they add this GTPase inhibitor, you get more fluorescence. The intensity is increased. And they say this is consistent with the idea that the drug is inhibiting the division of the cells, but DNA replication continues, so you get more DNA signal inside. When they use the Cipro inhibitor of DNA gyrase, they get a reduced fluorescent signal, and of course that's a DNA synthesis inhibitor. And in both cases, you get aberrant Y formation. So they conclude that the production of a new arm takes place before DNA replication occurs, but the separation of the two new bacteria requires the completion of DNA synthesis. So basically, what we have here, the Y, of course, is the bacterium dividing along the longitudinal axis. Most bacteria divide by binary fission. Of course, if you have a rod, the division point occurs roughly in the middle, and the two ends of the rod separate. They don't divide longitudinally as these do in this case. And there are other examples of bacteria where you find Y shapes like this, and which are presumed to divide longitudinally, like Actinomyces, Bifidobacteria, Rhizobium, and in the spiroplasma, other species as well. So it seems to be prevalent there. And that, of course, brings us to the why question. Why do they divide, which we can't ask in biology, but we can say what selective pressure would lead to this mode of cell division where it goes longitudinally. Because if I understand, I tried to look for this, spirochetes, Treponema and Borrelia, they don't divide longitudinally. They just break in half, right? So why do these bacteria do it? And they suggest maybe, so it's very important for these bacteria to be motile, and the motility depends on long contractile fibers that go longitudinally in the bacteria. And so maybe dividing longitudinally is a way to preserve these fibers so you don't have to cut them in half. But that's a hypothesis that they don't know, of course. And that's our snippet. I thought it was cool, Y-shaped bacteria. Yes.
I'd like to add something to it because there is another case of longitudinal division in bacteria where the why question can be answered, at least tentatively. This is work from Silvia Bulgharese's group in Vienna, Austria, and it's about a bug which lives on the cuticle of nematodes. It's a gamma protobacterium, also can be cultivated, etc., etc. Not much is known about it, except this bug sits on its end on the cuticle of the nematodes. And there are so many of them that they make a total covering. They cover the surface of the nematode. Just so imagine a palisade stuck on a nematode. Okay. Now, these guys divide longitudinally, but they do it from both ends.
Mm-hmm.
Unlike the spiroplasma, which seemingly does it from one end. And here, the choice between binary fission in the middle and longitudinal fission is obvious. If you divide in the way normal bacteria do divide, the daughter cells will fall off. They'll be gone. Mm-hmm. But if you divide longitudinally, both progeny cells will have an end that sticks to the nematode. So here, and also here, the FTSZ ring They studied it rather nicely. The FTSE ring is not across the cell like it is in all rod-shaped bacteria, but it's longitudinal also. So it's a long oval that goes from one end of the cell to the other with gaps, by the way, which is interesting. So the disposition so they know a little bit more about this bug than they do about the spiroplasma.
Well, maybe there's a part of the life cycle of spiroplasma in the insect where it's attached to something, and this would be important, right? That makes a lot of sense.
Yeah, that could be. But they don't know that. Whereas in the nematode, it's a little bit easier to study because of the geometric disposition.
Curiouser and curiouser. You know what that's from, right? No, it's from Alice in Wonderland.
Just remember, bacteria learn the long division.
That's right. All right. That was a snippet. And before we go on to our main paper, I want to tell you about one of our sponsors, CuriosityStream, which is the first ad-free nonfiction streaming service in the world. They have now over 1,500 titles, founded by John Hendricks of Discovery Communications. And it consists of real science shows, not reality TV. You can get it on many platforms, the web, Roku, Android, iOS, Chromecast, Amazon Fire, and Kindle, Apple TV, 196 countries. And what you get is a wide variety of science and technology content and nature, history, interviews, but it's all nonfiction. It's real science. In addition, they have interviews and lectures. They have Next World with Michio Kaku. They have a number of programs on viruses and bacteria, a program called Inside the Human Body, Microbirth, Leaps in Evolution, and much more. So I think this might be a good fit for our listeners. So you should check it out. Monthly and annual plans are available starting at just $2.99 a month, $2.99, less than a cup of coffee, at least here in New York City and probably throughout the country. I was reading this ad on TWiM the other day, and our guest was Harold Varmus. And I said, it's less than that cup of coffee you just had at Starbucks. And he said, Tom Maniata's bought it for me. I didn't pay for it. Okay. Dropping names.
How was that conversation?
Oh, it was just amazing. He's so smart.
Has it posted yet? Has that episode posted?
It has posted. And I mean, he is has such insight into everything you ask him. He has just an amazing career, incredible beginning and middle and current. And so, yeah, it was a wonderful conversation. Kathy came out here to join us for it. She flew out.
I will tune into that one.
Mm-hmm.
For curious minds who never stopped asking why, check out curiositystream.com/microbe. Use the promo code microbe during sign up to get unlimited access to the world's top documentaries and nonfiction series completely free for the first 60 days. That's 2 entire months free of one of the largest 4K libraries around. That's ultra high definition. Just go to curiositystream.com/microbe and use the offer code microbe at sign up. And we thank CuriosityStream for their support of TWiM. All right. Michele, do you have a charge for us?
I do. We are going to switch to the topic of electrobiology. And this is thanks to a paper titled Thermally Activated Charge Transport in Microbial Protein Nanowires. And it is published in the journal Scientific Reports in March of 2016, and it is a collaboration from colleagues in the Department of Microbiology and Molecular Genetics and the Department of Physics and Astronomy at Michigan State University and the Nanobionics group at the UNESP Institute in Sao Paulo, Brazil. And Elio, I would love it if you would actually read the formal name of the department, because I love to hear your voice when you're speaking your native tongue.
This is not Spanish, but Portuguese, so I'll try my best Portuguese.
Yeah.
Departamento de Físico-Química, Instituto de Química, Universidade Estadual Paulista Júlio de Mesquita Filho. That sounds a little bit Portuguese, doesn't it?
Yeah, very
Save
much.
your trip to the Olympics.
That's right. So the authors are Sanella Lampa Pesterk, Joshua Veazey, Kathleen Walsh, Gustavo Feliciano, Rebecca Stadel, Stuart Tesmer, and Hema Reguera. And they have been using as a model system Geobacter to understand electrobiology. And Geobacter is a gram-negative bacterium. It lives, or genus. These bacteria live in soil or the beds of lakes, oceans, and other bodies of water. And they get their energy by anaerobic respiration. So they can feed off acetate, which is an abundant carbon source that other microbes generate when they're fermenting organic matter. But they do this in a very weird and wonderful way, which is these Geobacter cells make long appendages, type 4 pili, that they use to attach to insoluble metals in the soil, like ferric iron or rust. They actually use this iron as the terminal electron acceptor as they undergo anaerobic respiration. How do they do this? So the bacterium are able to oxidize acetate and then transfer electrons across their membrane using membrane-bound proteins known as cytochromes. And then those electrons transfer onto the pili, which then are transported away from the cell quite some distance onto the terminal electron acceptor, in this case, rust. So in this paper, the group at MSU was able to measure the flow of electrons across these purified pili and then use structural biology and genetics to identify the path that these electrons took as they hopped along the pili onto the metal. So let me back up and give some background on just what respiration is. So usually when we hear this term, we think about breathing oxygen. Biologists also think of electron transport chains. So these are proteinaceous complexes in cell membranes that can pass electrons from one protein component to the next. They're built of proteins called cytochromes, C-type cytochromes. And then their final electron acceptor, in the case of aerobic organisms, is oxygen, and they generate water. Now, as they're passing these electrons, the cell is also pumping protons out of the cell. This generates the proton motive force, which a neighboring enzyme, the ATP synthase, can use to harness to convert ATP, to generate ATP from ADP. So by oxidizing organic compounds in the cytoplasm, the cells can generate ATP. Now, a number of microbes, many of them, live in places where they don't have oxygen available as the terminal electron acceptor, but they can also generate ATP using electron transport chains. But in this case, their terminal electron acceptor is an organic or inorganic compound. So for the Geobacter, they actually use metals, iron or uranium, in fact, as their terminal electron acceptor. We know from looking at the genome that the Geobacter are heavily invested in anaerobic respiration. They have more than 100 genes that encode cytochrome C-type proteins. Their cell envelope is rich in these cytochromes, but out in nature, they also form biofilms and actually have extracellular cytochromes that also contribute to this electron transport along with the pili. But it's the pili that are the focus of this biophysical paper. And so just to remind you, pili are fibers, appendages that many bacteria make. These happen to be 2 nanometers in diameter, and they self-assemble from a single protein, the pilin protein. So it's a very neat experimental system because you can just purify the pilin protein and then under appropriate conditions, it will assemble into these long appendages, which you can then purify and use in your experiments. So we know from many bacteria that pili are useful for mediating attachment of microbes either to surfaces or to other cells in the population. But here, Geobacter also use them to respire, to basically breathe using metals. Because pili are so widespread in the microbial world, we have a rich literature on how they're made, how they're assembled, how they're regulated, how they function. So this group was able to exploit that literature to do these really beautiful structure function biophysical experiments to ask whether the pili are capable of conducting electricity and at how much, what's the current?
Or by themselves, right?
All by themselves, pure.
All by themselves.
Yeah, so in this project, this paper, they're using purified pili and measuring their ability to transmit electrons. So how they did this was really technically very sophisticated and demanding. They used a technology called conductive probe atomic force microscopy. And basically the setup was they have an electrode, and then they spread across a surface their purified pili fibers so that some of them will be touching that electrode. And then their atomic force microscope has a probe which they can manipulate. So they would manipulate that probe to touch the pili at some distance from the electrode. In this case, anywhere from 50 to 900 nanometers. They then can apply voltage to the electrode, and they use the physiological range here of 0.1 to 1 volt. And then using their probe, they could measure current. So I know I explained that in only a few minutes, but I was able to talk to Sanella, the first author on this paper, who is the postdoc who did much of this work, and she convinced me that these were incredibly demanding experiments, very difficult. For example, this microscope, which imagine now this probe is touching this tiny pilus. It's very sensitive to vibrations. So Sanella found that she had to do much of her work late at night when everyone else had gone home.
Oh my God.
So it took a lot
Don't let anybody move, huh?
It took a lot of persistence and troubleshooting, but she described the late night where she actually, for the first time, measured current and was able to plot that as current versus voltage as a really exciting moment in this challenging project. So that data is shown in figure 1 of this paper. Again, it's the current versus voltage plot, and this is a standard way to measure and display conductance. So they were able to determine that these pili can transport electrons at the rate of 10 to the 9th electrons per second, which is 100 times faster than the canonical membrane-bound electron transport chains many of us learned about in our biology classes. And then for the aficionados out there who think about electricity, this translates to a conductivity of 1 to 4 Siemens per centimeter. Their next question, so having demonstrated that these purified pili, their purified pili preps were conducting electricity, they then wanted to ask, was it the purified pili made of just this self-assembled pillin protein sufficient, or was it instead acting as a scaffold? Perhaps some metals contaminated their equipment during the purification, and that some metal or other protein was contributing. So to address that, again, they used highly sophisticated methods, plasma atomic emission spectroscopy, to ask if they were contaminating metals, cytochromes, flavins, quinones, other components that are typical of well-characterized electron transport chains. But they found no evidence. They could detect no contamination with these other components. So they were able to move forward then, confident that it was the pili itself that was conducting the electricity. So they next wanted to deduce how the electrons were being transported along this proteinaceous fiber. And to do that, they studied the three-dimensional structure they have of the pili. And in particular, they were looking for amino acids that could bind and be a platform for hopping electrons. So in figure 3 of this paper, they show the structure and they highlight all of the tyrosines and phenylalanines. So these are aromatic amino acids that can serve as a platform for electronics to hop and pass from one residue to the other.
I must say, I had no idea about that. It really hit me between the eyes when I found it out. That's really amazing, isn't it?
That the tyrosines and phenylalanines?
That the aromatic amino acids are so good at conducting electricity.
Yeah, and these pili in particular have, you'll see from the paper, quite a few of these aromatics that are close to one another. In fact, they have the measurements here somewhere between 7 and 10 angstroms, I believe, 6 and 10 angstroms on average. So they identified one tyrosine that, based on the architecture they predicted would be critical for this electron hopping. And they changed that tyrosine to an alanine, which would then remove that member in this putative chain hopping surface. And they then repeated their experiments and measured the transfer of electrons from one part of the pili to the other using that same system. And in figure 4, what we see, again in another plot of current versus voltage, is that the wild type pili protein undergoes a transition from what is called, and this is getting a little technical now, goes from a transition of an electronic transport. First it starts out as an ohmic regime at low voltage and then shifts to an electronic transport regime. And that shift, which you can recognize as a shift from a linear array of the data to a exponential curve, occurs for the wild type at around voltage 0.5, but for the mutant that now lacks one of these aromatic residues at a key position on the filament, that shift doesn't occur until there's a higher voltage, about 0.6. So as the experimentalists described this to me very patiently today, the way we can think about that is the amount of voltage you have to apply is an indicator of how much force needs to be applied to drive those electrons down the path. So the mutant that lacks that critical aromatic residue requires more force, higher voltage, to get the same movement of the electrons.
By the way, the accuracy of the measurements are really phenomenal because they let you fit the data to the two curves in such a beautiful way. It's really, I found it kind of remarkable.
It is. And in order to do that, we should give a shout out to the number of data points on each of these plots, because I think that probably represents quite a lot of painstaking work.
You think?
As far as I can understand how each measurement was collected. So they then went on to study the impact of temperature on conductance, and they also compared the Geobacter pili to pili purified from Pseudomonas aeruginosa. And that information they present in figure 5 with a number of lovely transmission EMs and also more plots of current versus voltage that shows biophysical parameters which are going to be useful in future studies that seek to exploit this biological system in designing nanowires made from biological material. So before we talk about the implications of this work and potential applications of it, I want to return to the biology. What could be the benefit to Geobacter to having these pili that transmit electrons? So the group actually, Hema Reguera's group, actually published a paper just 2 days ago in Nature Communications, which looks at this in more detail. Now not studying the purified pili, but rather using genetics to separate and distinguish the contribution of the pili structures and the contributions of the extracellular cytochrome proteins and the biofilm extracellular matrix to the ability of Geobacter to respire and to transmit electricity. And what they're able to show really beautifully in that paper is that without the pili, Geobacter can still grow and form biofilms to a certain thickness. But in order to make a thicker biofilm, they need the pili to be able to transmit electrons. And again, they demonstrate that using this same tyrosine mutant that they characterized here. So they can interpret this to mean that the capacity of these pili to carry electrons away from the cell and donate them to rust or other insoluble metals in the environment allow Geobacter to form thicker, more luxuriant biofilms out in nature. So kind of exciting. It's very cool biology, but it's also exciting to think about how this living, genetically tractable microbial system could be exploited for generating energy in difficult places. So Hema shared with us the story of one application, which is people were able to bury an anode, metal anode, in the ocean floor and then wire it to a cathode that was floating on the ocean surface using floats. Within a short period of time, Geobacter naturally bound and formed biofilms on that buried metal plate. And they demonstrated that the Geobacter then transferred current along the wire up to the cathode that was floating on top. So this then provided a great opportunity to capture that current and use it to power sensors that marine biologists can use to track ocean life. So
they call
that's
this
just
a microbial fuel cell, don't they?
Right. Yeah, you want to tell us about that, Elio?
Not yet.
Not in more detail. Okay. So it's very cool that Geobacter, using just simple carbons like acetate that are very common in the environment, can then generate electricity if they have a place to deposit it. We could then capture that. And what makes this system really cool is that because it's biological, you can use genetic engineering to do the type of structure function experiments that they did in this paper to optimize certain features. Also, they point out that because this is a biological system, they don't need to use as many toxic solvents and metals as classic nanowire and nanotechnology currently has to use for their manufacture of these tiny transmitting wires. So that is the end of the paper. Do either of you have comments that you wanted to add before I talk about the first author?
Let me add something about the possible uses of this. Like you said, when metals get reduced, they tend to become insoluble, and this is why we have so much rust in the world, iron in the form of rust. But it's also possible to think about this in terms of bioremediation, specifically trying to get rid of uranium-6 metal in that form, which is abundant in as a result of making atomic bombs and so forth, is quite soluble and therefore hard to get rid of. But these bacteria convert uranium-6 into uranium-4, which is practically insoluble. And of course, if it's insoluble, you can just scoop it up and get rid of it. And so this is a use that is being studied. There's a group at Stanford working with a group at Oak Ridge, which uses puts together a big pool, a set of pools containing soluble uranium-6 and adding the bacteria to convert into uranium-4. What they point out is that in the real world, contaminated sites contaminated with uranium are also contaminated with other nasty things like solvents, toxic metals, and so forth, which would be not very good for Geobacter. So then in reality, you need to have some pre-treatment in the real world. But still, this may be a way to get rid of contaminated sites where uranium is a big issue. So that's interesting.
So you mean my next cell phone is not going to have Geobacter pili in it, right? So the basic problem, if I understand, is that when you move electrons, then you end up making a product that is not good, like rust. So that's a limitation of the technology.
No, actually, in this case with Geobacter, they use rust to donate electrons, and then they convert it to a more soluble form.
So they do the opposite,
which then can be used by other organisms. So they out in nature, they actually grow in a consortium with another microbe that then harvests the
Does this have
ferrous.
to do with microbial mining, like it's used on a large scale for obtaining copper from insoluble copper into soluble copper? I don't know how that works, but I think that's done on a large scale in Chile and other places where copper is mined.
Another microbe that does this also has these electrical conducting properties is Schwanella. So they have extensions of their outer membrane that also bind cytochromes and have been shown to transmit an electrical current. So this is kind of an exciting new area for me is understanding how we could exploit microbial structures.
Yeah.
But I read Schumanella does it a little bit differently in that it does not have pili, but it has extrusions of the outer membrane. It also secretes flavins, which shuttle the electrons back and forth. But it's a different mechanism. So there are at least two ways of doing this, the Geobacter way and the Schumanella way.
Yeah, and Geobacter seems to do both because they too secrete these cytochromes that are bound by the extracellular matrix, the kind of gooey matrix that holds together the biofilm. But then they also extend these pili even further and allow them to grow even thicker biofilms.
Right. By the way, the pili are known as nanowires, aren't they?
Yeah, that's the term that they've coined, which is very clever.
So you've probably heard of the use of M13 filamentous phages to construct batteries.
No, I haven't. Tell us about it.
Well, there's a group at MIT whose name is escaping me, but they use, so basically you can take the property of the filamentous capsid protein of these phages to form regular arrays to construct batteries. So they don't have any electrical properties in themselves, but you can construct the battery by making arrays with them and incorporating the right chemical substrates to hold a charge. And they can develop low-power batteries, which are largely based on this phage protein, and they don't have a lot of toxic components that batteries do at the moment.
Wow.
Angela Belcher is her name.
How stable are they?
Oh, that's a good question. I'm not sure that they are yet very stable, but they do store a charge, and they're working on them to try and figure out if they could eventually be used just like the conductance here is being used. Stability, of course, would be a problem or an issue. But in fact, there's a TED talk by Angela Belcher on this. I'll put a link in the show notes. Someday your phones might have phages in them. Who knows? They already have viruses, and now they'll have real viruses.
I'd be an early adopter of that technology.
So it's interesting that you have two different. I mean, the phage protein doesn't have any inherent electrical capacity, as do the pili, but they can be used to make an electronic device. So it's really interesting.
Wow.
Yeah.
Sweet.
You can't have a conversation in microbiology without bringing up phages, can you?
No. Nor would you want to, right?
Nor would you want to. That's exactly right. So I was reminded in thinking about this of the wonderful saying by Albert Szent-Györgyi, a Nobel Prize winner. I think he discovered vitamin C and other things. He said, biology is nothing but electrons looking for a place to rest.
That's it. It's all of it. That's right.
So I was able to speak with Sanela, the first author. Her last name is Lampa Pesturk, and she is from Belgrade, which is now in Serbia. As an undergraduate, she studied physical chemistry. She said it was a really challenging program. They had to master math, chemistry, and physics. She described it as really grueling, but as she looks back on it, she realizes that with that solid foundation she has in those three sciences, she can now apply that knowledge in so many different ways. So she's found it liberating. But her first interest was actually writing plays. She aspired to be a playwright, but in her country, only 2 students were admitted to the academy as playwrights each year. So she then switched her interest to physical chemistry, perhaps in part influenced by her father, who was a chemist. So after she got her undergrad degree, she came to Michigan State here in East Lansing and got her PhD with Warren Beck, who is a biophysicist. She studied protein folding and learned their ultra-fast laser spectroscopy, and she's an expert in that science. She does credit Warren Beck with teaching her to be bold and not being afraid to go outside of the physical chemistry to tackle more challenging biological molecules. She was really grateful for her training with him. In Hema Reguera's group, which was her second postdoc, she described what a complex project this was, and it brought together people, of course, from physical chemistry backgrounds, microbiology, genetics, very different fields, and they frankly, speak different languages. So she said it was a challenge to try to get them all together and keep everybody up to date and motivated. But she credits Hema Reguera, the head of the lab, as being like a really determined and driven scientist whose ambition was contagious. And she says it was that really kept the group together and moving forward. Currently, she is a senior scientist at the University of Rochester, where she's doing bioelectrochemistry with the chair of the chemistry department, Bob Kraus. They are, again, she's using ultrafast laser spectroscopy to analyze nanotubes and biological tubes to produce hydrogen. She also is going to be starting a full-time teaching position at Nazareth College, where she looks forward to encouraging students, especially young women, to pursue careers in science and in the physical sciences in particular. And she is going to continue research there using the Schiwanella model for studying electromicrobiology. She and her husband are the parents of 2 daughters, 8 and 10. So she has a full and interesting life.
My goodness.
Yeah.
And both of you, of course, know the PI on this paper as well, don't you?
That's right. Elio, you can talk first about because you introduced me to Hema.
I met her in Spain. Hema is from Spain, from northern Spain. And I think she either got a PhD or did a postdoc or something like that in Barcelona. Anyhow, I met her at the meeting and we liked each other immediately. Spoke a little Spanish together. And since then, every time I had a chance to meet her, I looked her up because I enjoyed her company. She did a postdoc with Roberto Colter and with... oh my goodness, his name escapes me right now, and big man in conductance. I'll come to me. Anyhow, she is both Michele and I had the pleasure of working with her on a textbook called Microbe, second edition, published by ASM Press, and she displayed an immense amount of knowledge and skill, as well as a great style of writing. So it was really most productive to work with her.
And I'll add that the textbook includes some very clear drawings that help to explain and will help our listeners visualize the biology that we described in this story today. So I'll include a figure from the textbook of this respiration mechanism of Geobacter.
Wonderful.
It was Derek Lovely that she did the postdoc with.
That's right. And she also contributes to your blog, Small Things Considered, right?
Mm-hmm.
Oh, that's just lovely.
So I'd like to add something about this study because it is interesting how it unites 2 concerns. Uh-huh. One is making electricity, therefore making sustainable fuels and possible bioremediation. This deals with both, albeit on a very ultra-microscopic scale right now, but who knows what will come of it. So it seems promising and it seems wonderful that people should study this now.
And we can grow bacteria cheaply, quickly, large amounts. So it could scale up. I could see it.
Right.
All right. Thank you, Michele. Nice.
My pleasure.
Nice work. I want to tell you about our second sponsor, and this is a new sponsor for TWiM. We welcome Drobo. Drobo was designed to solve 2 problems that affect your hard disk drives in your computers. First, hard drives get full, and secondly, they are mechanical and will eventually fail. And hopefully it occurs in that order. They get full and then they get fail. And Drobo solves these problems. They give you expandable storage. You can put as many drives in as you want. These are black boxes, basically, with a number of different bays, and you can put more and more drives in up to the capacity, of course. They're aware of how much capacity you're using on the drives, and they have lights on the front that tell you when they're getting full, and they use redundancy. Let's say you have one of these Drobos with 5 drives in it, and one of them or 2 of them fails. You can pull them out and put a new one in, and you don't lose any data because it's duplicated across the whole array. They have actually a proprietary array technology. You may have heard of RAID arrays, of way of combining different drives together. Well, this is different and more secure. It's very simple. We don't want to be the family IT guy. Simple to use. and simple to interpret. If you can understand a traffic light, you can use a trobo. They have many different products, and depending on what you want to do, you can pick a different kind of product. If you're into photography, there are solutions like the Drobo 5DT or the 5D. If you want a portable solution, there's a Drobo Mini that weighs 2 pounds and you can carry around with you. If you need a lot of storage, there are 4, 3, or 5 drive solutions that'll work. And here's the one that I think is cool for a laboratory. They have a version, the Drobo 5N, which is a network-attached storage system. You just plug it into the Ethernet and share your files. For example, I'm going to be doing that within my laboratory. So it's pretty cool stuff, whether you need it for home or for your laboratory. And if you're a listener of the Microbe TV series of podcasts, you can save $100 on a purchase of several of the different models, the Mini 5D, 5N, or 8 drive or 12 drive systems. So they have boxes that go up to 12 drives and put 12 different hard drives in, get a lot of capacity. You can go to drobostore.com, use the discount code Microbe100 to get $100 off on your purchase of any of these devices. We thank Drobo for the support of TWiM.
Sounds like we could house a lot of episodes of TWiM and TWiV on those devices.
That's a very good point, because at the moment I have lots and lots and lots of individual hard drives. And yes, that 12-bay unit, I could put a lot of them on. That would be great. I do save them all, because you never know. I don't want to throw them out. So I figure someday, right. And of course, when I'm gone from this world, my kids will go, "What is this stuff?" And they'll throw it away. Oh, well. I wanted to read just a couple of email here. We have a few minutes left on this TWiM. The first one is from William, who writes, "Hi, TWimmers. It's a nice late July evening here in Berkeley. 60 Fahrenheit, 15.5 C. No fog, but we have had a bit less than usual summer fog this year. I ran across this article, which is fascinating because it falls into the category of what you thought was obvious, in fact, is not. And it is an article in Science News, which is called Lichen. Apparently happy couple, really a threesome. Lichen comes in 2 basic flavors. One forms a thin film-like layer on rocks and trees. The other is composed of macrolichens that grow big leafy branching or vine-like structures. The latter seem to harbor a yeast. And this discovery started when a postdoc was studying 2 lichen species collected around the campus, in his campus in Montana, and they're distinguished by the presence of vulpinic acid in one, which gives it a yellow color. Genetic tests showed that the fungus and alga in both lichen species were identical. So they found the genetic signature of a third species, a basidiomycete yeast, present in both of the lichen species, but more abundant in the yellow version. They and their colleagues went on to test 56 different lichens from around the world and found each had its own distinct variety of yeast, basidiomycete yeast. So lichen is not just 2, it's 3 different organisms. How about that? Thank you, Bill. A letter from Anthony, who writes, "Let's celebrate," this is an article in The Guardian, "Let's celebrate 150 years of Beatrix Potter, author, scientist, and fungus lover."
I did not know that.
Oh, she did. She wrote a bunch of articles on fungal taxonomy and illustrated, and she has a lot of paintings, beautiful paintings.
I didn't know that, but Elio, you would know this because you're a mushroom guy.
Right.
So apparently this is an article all about Beatrix and her love for fungi. Thank you, Anthony. And the last one is from Christoph, who writes, "Dear TWiM overlords," parentheses, "in a gender-neutral way," the overlord, of course. Michael had a very interesting interpretation of what coccolithophores were when commenting on Kyle's letter in TWiM 131. It was our last episode. He correctly identified 2 out of the 3 Greek-Latin words in there, but that steered him in a funny direction, as he thought they were rock-eating cocci and not haptophyte algae. Let's see what I can remember from learning 2 dead languages in gymnasium, German grammar school. Coccolithophore.
High school.
He wrote grammar school. It's high school?
High school.
Gymnasium? All right. Coccolithophore consists of kakko from Greek kokos, which is berry. Michael got that right. Litho from Greek lithos equals rock. Score, Michael. And phore from Latin fere, to bear, or to carry. This one Michael got confused with trophos, or feed. So Michael turned the cocci rock bearers into cocci rock eaters. Close, but no cigar. It's great. Indeed, the cocci rock bearers got their name from carrying tiny scales of calcium carbonate. The coccoliths, I presume this was close enough to be called rock, since their fossilized remains make those impressive cliffs of Dover in the UK. Their ability to turn carbon dioxide into calcium carbonate, combined with their high abundance in temperate oceans, makes them highly important to the global carbon cycle, but unfortunately also highly susceptible to ocean acidification. One of their prettiest members, Emiliana Huxleyi, has even been featured on TWiM before. That's right, TWiM number 34. Hope I didn't screw this up too badly and embarrass my former Greek and Latin teachers. Keep up the great works. The Twix Empire is awesome. Maybe you could feature more free-living eukaryotic microbes and their viruses. It seems to me like they got lost somewhere in the gap between TWiM and TWIP, but their rare appearance is always a delightful listen, like TWiM 94. Chris, P.S. The weather in Vancouver, BC, is currently 21C, 73% humidity, 0% chance of precipitation, and apparently pretty windy with 29 kilometers per hour, although a quick look out the window can't confirm this. Thank you, Christoph. That'll do it for TWiM 132. You can find it at iTunes and also microbe.tv/TWiM. Consider becoming a patron of the TWiM and Microbe TV family of science shows. You can contribute as little as $1 a month at patreon.com/microbetv. And of course, as always, send your questions and comments to twim@microbe.tv. Michele Swanson can be found at the University of Michigan, which is in Ann Arbor, Michigan. Thank you, Michele.
Thank you. Great to be back.
Elio Schaechter is at Small Things Considered. Thank you, Elio.
Well, it was distinctly my pleasure. Always is.
And also, you can check out Elio's new book, In the Company of Microbes. It's a compilation of blog posts from Small Things Considered. It's published by ASM Press, which I got a signed copy at ASM Microbe. I treasure it. It'll be worth a lot of money in a few years. I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Microbiology for their support of TWiM, in particular Chris Condayan and Ray Ortega for their technical help. I'd also like to thank our sponsors, CuriosityStream and Robo. The music you hear on TWiM is composed and performed by Ronald Jenkins. You can find his work at ronaldjenkins.com. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.